All right, so the bacterium does not always have an opportunity to consume glucose. So how do these different pathways work? Which ones are used if the cell is starting with something other than glucose? So this is a figure from your text, and we're going to go through a couple of examples, but I don't know how well you guys can see this. This is pyruvate right here, and this is acetyl-CoA. And so you'll see how many arrows come to pyruvate and acetyl-CoA. So once you get acetyl-CoA, then the compound will go into the Krebs cycle, and that all happens pretty much the same way. But the different ways that each of the different nutrients get to acetyl-CoA can be important. But acetyl-CoA is a really, really common catabolite for different types of nutrients. So if the cell is consuming a carbohydrate, then it will use one of these different processes, which you do not need to know, to make glucose or fructose, which you do need to know. So all carbohydrates will be converted to glucose or fructose, and then will be run just through one of the different glycolysis pathways. If the cell is using a protein as its food source, then the first thing that will happen is enzymes called proteases will break the giant protein molecule down into individual amino acids. As a side note, the starvation media that you guys use in lab is glucose and peptone. And so proteins are giant and very globby, right? And so it is actually really difficult for bacteria to import whole proteins as a food source. So what we feed them almost always is peptones. And so peptones are partially digested proteins that are really, really easy for the bacteria to import and use. And so we feed them that in the starvation media. It's 1% glucose, 0.1% peptone. Also, the TSA that we feed them, tryptic soy agar, is soy proteins that have been partially digested by the enzyme trypsin. And so, just FYI, bacteria normally start with I've just forgotten the word, peptones, instead of with whole proteins. In either case, they still need to use proteases to break the protein/peptone down into individual amino acids. So then they've got their amino acid, and it is going to look like this. So the first thing that happens once the cell has individual amino acids is that this amino group here needs to come off. And so the pyruvate oxidation and the Krebs cycle are carbon-based systems, and the cell does not really have a great way to manage this nitrogen. And so it just has to get the amino group off. There are a couple of ways to do this. You just cut it off, and it leaves the cell as waste ammonia. So if you had a bacterium either in the urea agar or in one of the phenol red broths that turned hot pink, your bacterium was breaking down protein and releasing this amino group as ammonia. And that would raise the pH, and phenol red is hot pink at high pH. So option 1 is called deamination and is just straight up the removal and loss of the amino group. The second option is called transamination. And so transamination, you can see down here, and this is where the amino group is passed from one amino acid to a carbon skeleton, in this case alpha ketoglutarate, giving you a different amino acid and then another carbon skeleton. So instead of, we will learn eventually, actually farther on in exam 3 when we get to the nitrogen cycle, that making this amino group is incredibly energy intensive. It uses tons and tons and tons of ATPs. And so if the cell has available another carbon skeleton where it can just pass the amino group and not have it just be lost to the greater world at large, then it will do that. And so transamination is also very common. If your bacterium is subsisting entirely on protein, then it is releasing so many of these amine groups that they will just go out as waste ammonia. But just be aware that transamination is also an option, and it is the passing of the amine group. So once you've gotten rid of the amine group, the carbon skeleton will be further processed and will go into the Krebs cycle. And so then it will be completely oxidized to carbon dioxide, and we will be making some NADHs and some FADH2s and some GTPs. Again, remember, you do not need to know the numbers for any of those. All right, so what if the bacterium is eating a lipid? So that's also an option. We did not look at this in lab because it is bizarrely tricky to measure, but it does happen in real life. So if the bacterium is eating a lipid, then the first thing that it does is use enzymes called lipases to break down the lipid into the glycerol part and the fatty acid part. So the glycerol part gets converted to something called dihydroxyacetone phosphate. And this is a diagram of EMP glycolysis, the Embden-Meyerhoff-Parnas. And you can see this little box right here. If you were to, I'm not going to zoom in, but if you were to zoom way in on that, you would see that that box is dihydroxyacetone phosphate. So the lipase breaks the glycerol off. The glycerol immediately enters into glycolysis, and then from there, a pyruvate is generated, which goes to pyruvate oxidation, and then the Krebs cycle, eventually ending us up with carbon dioxide, NADH, and FADH2, and some number of ATPs/GTPs, depending on the final electron acceptor. So then, once the glycerol is off, we are left with the fatty acids. And so fatty acids are broken down through a process called beta oxidation. And so I am not a biochemist by any stretch of the imagination, but I have been told that even chain fatty acids are more common than odd chain fatty acids because both fatty acid assembly or anabolism and fatty acid catabolism happens by 2 carbon pieces. And so I understand that even numbered fatty acid chains are more common. And so I don't know if you remember or not, but acetyl-CoA is 2 carbons, right? So in pyruvate oxidation, we start with pyruvate, which is 3 carbons. We break off a carbon, it goes out as carbon dioxide. We are left with 2 carbons as acetyl-CoA. And so in the process of beta oxidation, the fatty acid chain is broken apart 2 carbons at a time, and acetyl-CoA is produced. It will usually go to the Krebs cycle. Sorry, wrong way. This scroller wheel thing is backwards. Anyway, the acetyl-CoA will usually go to the Krebs cycle, where again, broken down to carbon dioxide with production of NADH, FADH2, and GTP. But there may be some times, remember I told you that glycolysis, pyruvate oxidation, and the Krebs cycle are themselves catabolic processes because they break down things, but that they will support anabolism. And so the acetyl-CoA that is produced either by beta oxidation or through pyruvate oxidation can also be used for anabolism to build stuff in the cell. The beta oxidation of fatty acids produces acetyl-CoA. It also produces NADH and FADH2 because bonds are being broken, and so those electrons are captured and will most likely eventually make their way to the electron transport chain.